So this study is also very interesting because it looks at changes that we think happen in models, in cells, in mice, and we think in humans or in the human brain as well, when they accumulate alpha-synuclein. So what we looked here was more at not at what triggers alpha-synuclein accumulation, like glycation or other factors, but more what happens when alpha-synuclein accumulates and becomes toxic. And one of the things we found that is happening is changes in gene expression. And this means changes in the levels of a molecule that is called a messenger RNA. And then we started looking at other RNA molecules, and there's a class of RNA molecules called the small RNAs or microRNAs, and we found that there were some microRNAs that were upregulated, that the levels were increased, and others that were decreased. And because these molecules regulate the expression of some messenger RNAs, we could find a correlation between the two. And what was striking was this was pointing us in the direction of synaptic function or dysfunction. And so in this study that my PhD student, Mary Shilaki from Greece, did, was to study this process and to validate some of the targets that we found. So we did complicated bioinformatic analysis, and then we selected some hits that were involved and related to synaptic function, and she did some studies to validate some of the findings, and she indeed found that there were some proteins involved in synaptic biology that were changing because of the accumulation of alpha-synuclein. So it's something that we are still continuing, but the results are looking really exciting. So we're very curious to see where this will lead us, because maybe the hope is that this will give us ideas about possible targets for therapeutic intervention.